105's command. Hello, Dr. Long here. Today we'll be going over SCP-047. Let's get right into it. SCP-047. Object Class: Kether. Special Containment Procedures. SCP-047 is to be contained in a 0.5 meter by 0.5 meter by 1 meter hermetically sealed storage box at all times. This box is to be locked in Storage Locker 047-A inside P3 Secure Biohazard Lab 047-B. Any entrance to and activity inside 047-B will be recorded by biometric scan, closed-circuit cameras, and redacted. Entry to 047-B required the authorization of the project manager in addition to at least one O5 level clearance. SCP-047 is to be treated as priority 4 contagious biohazard in all protocols, including mandatory quarantine if exposed. Site Q047 has been provided, adjunct to Lab 047-B for this purpose. In the event of outside contamination of SCP-047-1, Lockdown Protocol 047-1, Yersinia, must be engaged. Description: SCP-047 is a heavily rusted breech gas cylinder made of an iron redacted alloy. When exposed to open air, the material of the cylinder evaporates slowly, producing a previously undocumented mutagenic gas. This gas has no effect on eukaryotic organisms, e.g. humans, but profoundly alters prokaryotes. Showing preference for common human microbiota, the natural microorganisms that live in the skin and throughout the body. On rare occasion, mutation produces a superbug, collectively known as SCP-047-1, a natural commensal with enhanced survivability and therefore opportunistic pathogenicity. The pattern of changes induced by SCP-047 suggests that these highly infectious microbes are at least to some degree selected for. Although the specifics of SCP-047-1 species are dependent on the base bacterium from which it is derived, there are several characteristics which appear to be generally consistent across all cases of SCP-047-1 mutation. Enhanced survivability in bacterium's natural environment and similar environments. Full spectrum antibiotic resistance. Increased reproduction rate and consumption of available materials. Development of sporulation ability in gram-positive bacteria. Increased ability to uptake, hold, and share plasmids, particularly in gram-negative bacteria. Increased transmission due to traits described above. SCP-047-1 samples are normally debilitating and virulent. However, compared to other Kether class SCPs, it should be noted that SCP-047-1 have a relatively low mortality rate due to their action through mundane biological pathways. Several strains of bacteria have been selectively mutated by SCP-047. Mutation of bacteria in culture is possible, but the process appears to be much more effective with bacteria living on a human host. Generally, mutation of natural commensals for experimental purpose is encouraged. After the containment breach of January 30, 2010, see Incident Report Yersinia 0471-01 2010, the mutations of already pathogenic species is banned and all existing samples must be destroyed. 3 particular species of SCP-047-1 mutated bacteria are of note, due to their involvement in containment breach of 2000. Propionibacterium 047-A is a strain of Propionibacterium acne mutated by SCP-047. Pathogenicity: severe skin colonization around sebaceous glands, modification to skin pH to levels that become toxic to the cells, massive inflammation and immune cell infiltration, eventually breakdown of skin structure leading to sepsis. Transmission: transmitted by skin-to-skin contact. Remain active on inorganic surfaces for up to 5 hours. Lethality: approximately 40% mortality rate. runs its course in 2 to 6 weeks, very visible symptoms within 5 to 10 hours, contagious within 2 to 5 hours. Handling. As soon as visible symptoms form, victims must be quarantined. Deceased victims should be incinerated. Streptococcus 047-C is a strain of Streptococcus mitis mutated by SCP-047. Pathogenicity causes inflammation of the mouth and esophagus initially, leads to open sores in the mouth, which result in S047C entering the bloodstream and becoming septic. Death is usually due to infectious endocarditis. Transmission, droplet, can remain active indefinitely by sporulation. Lethality, approximately 35% mortality rate, may become a recurring chronic condition if non-lethal. Handling. Subjects with any signs of mouth infection should be quarantined. Deceased victims should be incinerated. Clostridium 047-A is a strain of Clostridium difficile mutated by SCP-047. Pathogenicity unknown. C047A was developed from tissue culture and has never been exposed to a human. No samples remain in Foundation control. Transmission unknown, presumably transmitted through fecal contamination as with C. difficile. Due to smaller, more robust spores, may also aerosolize with flatus. Effects of aerosol intake of C047A cannot be predicted. Lethality unknown. Presumed extremely high risk of destruction of endothelial lining of gastrointestinal tract, leading to inflammation, sepsis, and toxic megacolon. Handling. Until further research has been done, victims should be quarantined and placed under 24-hour medical observation to develop functional diagnostic for this strain. Deceased victims should not be incinerated until adequate etiological research has been performed. Recovery Log 047. SCP-047 was recovered from Site Secure Laboratory by Foundation Biohazard Recovery Team in response to a full compromise situation on 1990. Testing logs indicate that the research team was attempting to contain data expunged in a Class SCP stable pressure cylinder, which led to combining with a full molecular biological analysis of this is available in Redacted. The initial release of gas when SCP-047 was structurally compromised was sufficient to create a microbial bloom of unaccounted species of SCP-047-1, killing all staff in the lab within blank hours. Exposed site staff obeyed standard Foundation quarantine/containment protocol, and the infection was contained successfully. End of audio log. I'll now read Incident Report Yersinia 0471-2010, and that's going to be it for today. I'll see you folks in the next one. Incident Report Yersinia 047-1 2010. SCPs involved SCP-047. Description: on January 30th, at approximately 3 a.m., Storage Locker 047-C, Site Lab 047-B, containing bacterial samples mutated by SCP-047 was compromised after a complete simultaneous data expunged, leading to a failure of security measurements in the area. Three samples of a total 12 were stolen. Data expunged since the initial containment break. Outbreaks of one of the stolen bacterial strains, Propionibacterium 047A, were recorded globally in communities of increasing size and population density. Further information on the stolen material, spread, and containment follows. Compromised items: Propionibacterium 047A, Streptococcus 047C, and Clostridium 047-A. See SCP-047 for further information. Outbreak information. First outbreak, P047A, February 27th, 2010. Siberia. Contained. See Incident Report P047A-03, 2010. Second outbreak. P047A. March 30th, 2010. Northwest Territories, Canada. Contained. See Incident Report P07A-04, 2010. Third outbreak. P047A. April 29th, 2010. South Australia. Contained. See Incident Report P047A-05, 2010. Fourth outbreak. P047A. May 27th, 2010. Mato Grosso, Brazil. Belief contained. See Incident Report P047A-06, 2010. Warning. Agents in the area are advised to familiarize themselves with the symptoms of P047A and be on the lookout for possible infection. Fifth outbreak. P047A. June 26, 2010. Redacted, Iraq. Site immediately data expunged, which is believed to have contained the infection. Access to incident report denied without O5 clearance. Sixth outbreak. P047A. July 26, 2010. Cameroon. Quarantine enacted. Efforts to track outgoing civilians underway. Infection not contained. See Incident Report P047A-07, 2010. Seventh outbreak. P047-A. August 24, 2010. Dalarna, Sweden. Quarantine enacted. Believed contained. See Incident Report P047A-08, 2010. Warning. Agents in the area are advised to familiarize themselves with the symptoms of P047A and be on the lookout for possible infection. 8th outbreak. Not recorded. Believed to have been taking place in North Korea. Data expunged. Agents with government access are attempting to gain access to parallel information. But due to data expunged, local services have been extremely uncooperative. Containment status unknown. 9th outbreak. P047A. October 23rd, 2010. South Carolina, United States of America. Quarantine enacted. Efforts to track outgoing civilians primarily successful. One civilian in a pickup truck is believed to have data expunged. Infection not contained. Resolution. Reports from data expunge indicate no further outbreaks are believed likely, but agents are advised to be on the lookout for new flare-ups resulting from uncontained civilians in previous outbreak regions. These may continue for years to come due to P047A's sporulation. Investigation into the cause of the initial compromise is underway. Anyone with useful information may anonymously contact the security via the attached form.